Trying to share the PowerPoint. Let me know if you can see the screen. And the PowerPoint. Can you see anything? Screen, PowerPoint?
No, sir.
No, sir, not yet.
Okay.
Yes, sir.
Can you see the screen right now?
Yes, sir. Yes,
Yes,
sir.
sir. It's visible.
Hopefully you'll be able to see the PowerPoint as well. Can you see the PowerPoint?
Yes, sir.
Yes, sir.
Sir, attendance form.
Okay. That will be given in letter. So I'm going into full screen mode and then I will start. Something weird is happening. Can't see the PowerPoint. I suppose you also cannot see. So we will be discussing, we're in the middle of discussing about the cells and organs of the immune system. Okay, so in the last class, we discussed about immune organs. We categorized them into two different parts known as primary lymphoid organs and secondary lymphoid organs. We say that in case of primary lymphoid organs, we have two different types of organ mostly, that is our lymph nodes and also the thymus. And the bone marrow and thymus, where we found that the immune cells, the lymphocytes, mostly they get matured there, and that's why it's called primary lymphoid organ. On the other hand, for secondary lymphoid organ, mostly we have three different types of secondary lymphoid organ: lymph nodes, spleen, and Peyer's patches. Those are the areas where the immune cells get the chance for the first time to encounter the antigens, different types of microbes. Today, we'll be focusing on the cells of immune system. I still didn't get the full screen mode here. So let's continue in this way. So cells of immune system. Please confirm whether my slide is moving or not. I just moved from, okay, now it's got this. Is it moving?
No, sir.
Okay, so still in the first slide, cells of immune system. Okay. So let's continue. Hopefully it will come. So we said that in case of blood, if you look into the blood, it has four different types of components. One is red blood cell, another is platelets, third number is white blood cell, and then the plasma, that is the aquatic part or watery part of the blood. And we also mentioned that red blood cell and platelets, they do not have anything to do with the immunity. It is not their function. They have different type of defined functions. And also, of course, the watery part has no role to play in immune system other than to carry the immune cells. So whatever immune cells are is actually white blood cells. White blood cells are the immune cells, although that is not the only one type of cells. Now let's see what different types of white blood cell we have. White blood cell has a different name as well. It's called leukocytes. Leuko means always is white. That's why we sometimes use leucoplast for bandaging purpose, etc. Those are also white in color. So leuko means white. So leukocytes. Red blood cells are known as erythrocytes, whereas WBC are known as leukocytes. Leukocytes could be broadly categorized into two parts: granulocyte and agranulocyte. Granulocyte, the name came from the granules. So if you look here in this type of cells, you will see, if you see under the microscope, all different these cells will show some presence of granules. ছোট ছোট বালির দানার মতো কোনা cytoplasm. In case of inside the cytoplasm, you will be able to see the granules. But here we do not have any granules in the cytoplasm. Okay. So granulocytes, the Bengali is danadar, and agranulocytes are adanadar. So granulocytes and agranulocytes. So granulocytes could be subdivided again into three different types of white blood cell known as neutrophil, eosinophil, and basophil. I'm pretty sure that you have heard about all these types of cells and in your higher secondary classes or for different other reasons. Now, in case of agranulocytes, we have two kinds of cells: lymphocytes and monocytes. So 3 under granulocytes and 2 under agranulocytes: neutrophil, eosinophil, basophil, lymphocyte, and monocyte. First, let's see if you have recently tested your blood, you or some of your family members. Two types of blood test is very commonly recommended by the doctors, known as TC and DC. TC means total count and DC means differential count. Total count means how many numbers of total RBC and WBC are there in the per ml volume. And differential count means, tell me what is the number of different white blood cells: neutrophil, eosinophil, basophil, monocytes, and lymphocytes. Why these are required, we'll try to understand here also. Neutrophil is, you can see all these, as I said, are immune cells. So they must be in our system in a more or less fixed number always. If you are a healthy person, when there is an infection, so you are suffering from some kind of infection, then the possibility are there that this immune cells number will increase. That means more and more immune cells are being produced to combat, to fight against the infection situation. So maybe apparently you are suffering from, somebody suffering from some kind of fever. So it's not pretty clear whether this is a fever due to some kind of cold or anything, or maybe due to some microbial infection. So if you do a blood test like differential count, then that can give the medical practitioners, the doctors, an idea whether this person is going through an infection or not. Okay. Now, generally, for a healthy person, the maximum number of white blood cell that is found in our blood is neutrophil. About 60% on an average, 50 to 70% is a range. The second one is lymphocyte. So one coming from granulocyte part, neutrophil, 60%, and then 30% is lymphocyte. A range is about 20 to 40%. The other three, eosinophil, basophil, and monocytes, their percentage is pretty low. For monocyte, it's more or less okay, 5% average range. but eosinophil and basophils are very less. So sometimes if you look into the blood test result, you might see that eosinophil is a 0, basophil is 0. So you may wonder that why I do not have any eosinophil or basophil in my blood. That is not true. There are eosinophil and basophil in your blood, pretty sure. But what happens when the laboratory technician counts this blood sample or the blood cells under the microscope? He generally takes 3 to 4 different types of fields and give a average value. So most probably he didn't find anything like eosinophil or basophil in those 4 fields, and that's why he said is 0. And 0 for a eosinophil or basophil value for a normal human being is acceptable. It is not indicating any disease condition or anything. Rather, on the other hand, if we see a huge amount of eosinophil or basophil in your blood, then that is alarming. That indicates that something wrong is going on. Okay, so this is the differential leukocyte count. Follow?
স্যার অ্যাসিডোফিল বলে কিছু হয়?
অ্যাসিডোফিল বলে কোনো ব্লাড সেল হয় না। অ্যাসিডোফিল হচ্ছে এক ধরনের মাইক্রোবস। ঠিক আছে? অ্যাসিডোফিল হচ্ছে who likes or loves acid, so who survives in a high acidic condition or very low pH. There is no relation of acidophils with the blood cells. Okay, fine. So differential leukocyte. So now we will focus into the lymphocyte. We'll come to the neutrophil maybe in the later part and later class. Lymphocytes. The most important thing, if you remember what I discussed in the last 2 class, that when 2 school of scientists were fighting kind of thing, that how immunity could be transferred. So one group of scientists was saying that something is antibody is there and that is being transferred through the serum. So that's what's called humoral immunity. Then Eli Metchnikov says that no, some cells are also very much responsible. So that is cell-mediated immunity. Then later it was found, well, there is a cell, one type of cell known as lymphocytes, which can link between these two, humoral immunity and cell-mediated immunity. And later we found that there are 2 types of lymphocytes, T and B, etc. So lymphocytes, as you have seen, just 30% or more than 20%, 20 to 40% range lymphocytes are there, white blood cell. But in case of lymph, 99% of the lymph is actually lymphocytes. Okay, so on the blood, 20 to 40%, but in case of lymph, 99% is lymphocytes. And at any given period of time, we have approximately 10 to the power 11 number of lymphocytes in our human body. 10 to the power 11, that's a huge number of lymphocytes. So what they're doing, they're continuously circulating in the blood and lymph. So like the patrolling security guard, continuously going in every nook and corner of our body, circulating through blood and lymph. Also, not only that, they're capable of invading or going into the tissue space like this. Okay. And so what is the purpose? They're trying to find out if there is any microorganism so that they can encounter. Okay. So that's why huge number of lymphocytes are there, which is trying to secure your safety. You're trying to save you from microbial infection. So you see, as I said, every time we are encountering thousands and thousands of different types of microbes. Microbes are everywhere, starting from the deep down in the soil to the deep in the ocean to the atmosphere in the hard kilometer, 10, 20 kilometer up to that range. Everywhere is microbes. So we are kind of swimming in the microbes all the time. Okay, but we are getting seldom sick. We're not getting infected, or maybe we are getting very mild infection, more or less. So that is thanks to this type of cells, as they're on the background continuously working for you. They're working like a patrolling security. Fine. Now, these lymphocytes, at one particular type of cell, there is no match with any other cell in our system, which has some important attributes. Attributes means characteristics, known as diversity, specificity, memory, self and non-self recognition. So gradually we will try to understand the actual meaning of this thing, but let's here discuss a bit. First, diversity. That means lymphocytes are not only one type. There are different types of lymphocytes. We know about T and B. We'll see one another in a while. Number two, specificity. Specificity is a very important term here. So the lymphocytes are very specific for microorganism. I'm not elaborating here. I will come to this point in a while. Number three, memory. Only one kind of cell which I can say has some memory. Okay, so we cannot say this about any other type of cell which possesses some kind of memory. But do not compare this memory as our memory. These cells do not have any brain or anything like that, so they cannot remember anything. What is the meaning of this memory? That is also we will see in a while. Number four, very important thing, self, non-self recognition. If you remember, at the very first we have seen that immune response have two different main attributes. One is recognition, another is response. So recognition, that means lymphocyte can recognize that which is actually my own cell, human body cell, and which is not human body cell. That's why you can distinguish between them and they can react it that way. So four different attributes of lymphocytes: diversity, specificity, memory, and self and non-self recognition. So we only are talking about lymphocytes here. So all other type of immune cells or white blood cells like neutrophil, eosinophil, basophil, monocytes, what about those? They do not have this kind of property, but they are playing some accessory role. So principal role are being played by lymphocytes, which are type of agranulocyte type of white blood cell or leukocyte. All other type of cells, the four different kinds of white blood cell, they are playing vital role, but accessory role in terms of immunity, like engulfing, destroying microorganism, so on. So, okay. So this lymphocyte, as I'm saying, could be categorized into three different types. So far, we have only seen about B and T, but there is also another one called natural killer cell or NK cell. So B lymphocyte, also known as B cell, T lymphocyte, also known as T cell, and natural killer cell or natural killer lymphocyte or NK cell, as we say them colloquially. Okay. So now the question is how you are distinguishing these cells. What is the criteria to be a B cell, to be a T cell, and to be a natural killer cell? The main thing is, of course, their function. Functionally, they're different. B cell, T cell, and NK cells are
স্যার আপনি মিউট আছেন স্যার।
শোনা যাচ্ছে এখন?
Yes, sir.
মিউট
Yes, sir.
করবে।
Yes.
আচ্ছা, দেখছি আবার। Okay, so we are in this slide. So we're asking that how we have distinguished these lymphocytes based on function. That means B cell, T cell, and natural killer cell has different functions. And another one is their cell membrane component. So I was saying that we already know from our school level knowledge that cell membranes are very much alive component, have a basic structure, protein, lipid, lipid bilayers are there. But not only that, cell membrane also contains different types of proteins, okay, different types of molecules. So based on that, we can say this is T lymphocyte, this is B lymphocyte, and this is NK. That means T, B, and NK cells are different in terms of their cell membrane component, and also they are functionally different from each other. So three different types of lymphocytes we have seen. Now, if we focus on the B lymphocyte or B cells or T lymphocytes, we will come to the NK cells later separately. B and T lymphocytes that not interacted with the antigen yet, referred to as naive. So maybe you have heard about this term naive. We usually use this term as a synonym of a very honest type of person, or they do not have much knowledge about anything. So say if somebody says the politicians are very honest, then we can say, oh, you are so naive about the politicians. That means you do not know anything about the politicians, something like that. Here also, B and T lymphocytes that are yet to interact with antigen, they have originated from, B cells from the bone marrow, T cells from the bone marrow as well. Then B cells is in their bone marrow, they're getting matured there. T lymphocytes migrate to the thymus, matured there, but they're matured, everything is fine, but they never get they're yet to get any chance to interact with antigen. Those are known as naive or unprimed B lymphocytes or T lymphocytes. And in case of B, there's also another name they got called small lymphocyte. So small lymphocyte is nothing but a naive lymphocyte, naive B lymphocyte. And they're small in size, they're motile, and they are non-phagocytic. We have seen a small clip in the last class about the phagocytic cell, how the cells are ingesting microorganism to the process called phagocytosis. So they are not phagocytic. They cannot digest or ingest any microorganism. They are very small. They're motile, known as small lymphocyte. Not only that, in case of small lymphocyte, another very distinguished feature, when you see under the microscope, it's possible to distinguish that. In case of small lymphocyte, if you see, the entire inside region is covered with nucleus. It's a huge nucleus like this. Okay, cytoplasm is very less. So cytoplasm is to nuclei volume ratio is pretty low, because we are putting the cytoplasm in the numerator. So cytoplasm volume is very low. So cytoplasm is kind of a rim around the nucleus. This is the distinguishing feature of small lymphocyte. They are naive lymphocyte or unprimed lymphocyte, and generally they have a short lifespan. That means they'll be produced, they'll be matured, and then they will die within a short period of time. Now, when they develop, these lymphocytes enlarges. Okay, and now they got a different name known as lymphoblast. So what is the difference between lymphoblast and B cell or small lymphocyte? Characteristic difference, first you can see, now it's become enlarged and the cytoplasm got a huge space. Nucleus no longer is covering the entire area. That means the cytoplasm is to nucleus ratio has increased. So higher cytoplasm and nucleus ratio. Another thing is inside organelle is become much more complex. Now we can see mitochondria. Now we can see the Golgi bodies or endoplasmic reticulum all over now there. Those are barely present in case of the naive B cell or small lymphocyte. So small lymphocyte develops into lymphoblast and showing this type of characteristics, higher cytoplasm nucleus ratio and more organelle complexity in comparison to the small lymphocyte. So here we are talking about the B cell mostly. We will not talking about the T cell, mostly the B lymphocytes. Okay. Now this lymphoblast, they have the capability to later even more proliferate and then to differentiate into two kinds of cell known as effector cell and the memory cell. So it's not that small lymphocyte. Lymphoblast is proliferating and differentiating into effector cell, also known as plasma cell, and the memory cell, the memory we are talking about. Lymphoblast differentiating into two different types of cell, effector cell and plasma cell, effector cell or plasma cell, and the memory cell. How that is happening, we'll gradually try to understand that. Let's see about the plasma cell. Plasma cell is very important. So plasma cell, as you can see, is nothing but a derivation of lymphoblast. And lymphoblast is coming from naive B cell or small lymphocyte. What lymphocyte talking about? The B lymphocyte. Okay. And we know the lymphocyte is one type of agranulocyte. Now, this plasma cell is important because these are antibody secreting cells. That's why it's called also effector cell. So who will secrete the antibody? Plasma cell will secrete the antibody. The effector cells also is called soluble antibody and thereby helps to eliminate different types of antigen. Okay. And these cells also has small lifespan. That means they will be converted to plasma cell, they will secrete some antibody, and then they will die. And a new set of cell will take place. On the other hand, another type of cell is memory cell. The persistence of this population of cell is responsible for lifelong immunity to many pathogens. This cell has a very, very long lifespan. Okay, and that is the mystery of having lifelong immunity. So we are getting vaccinated at the early years. Say in more or less in India, we take the vaccine dose or the booster dose at the age of 12. That is the last by mostly very early period after the birth, and then 5 years, and then 12 years. Maybe some of you still remember your last booster dose or something like that, and then never again. So you are going to, all of us are going to survive 70, 80 or 90 years maybe, but you are not taking any booster dose or anything like that in between, and you are quite confident that you are protected against those diseases such as mumps, measles, rubella, diphtheria, whooping cough, etc., and the TB, of course. So that lifelong immunity you are getting is due to these memory cells. Memory cells, they looks like a small lymphocyte. There is not much difference, but there is one major difference is there in terms of small lymphocytes and the memory cell. That memory cell contains certain kind of cell membrane molecule. Those are absent in the small lymphocytes. So under the microscope, it sometimes could be difficult to understand whether this is a small lymphocyte you're looking at or the memory cell. But if you do some biochemical study or other type of study, then based on the presence or absence of some kind of cell membrane molecule, you can say for sure that, well, this is a memory B cell or a memory cell, or this is actually a small B lymphocyte. Okay. So once again, say if we have seen the types of lymphocyte, B, T, and NK cell, then we're talking about the small lymphocyte, its characteristics feature, how small lymphocytes is then converted to lymphoblast and its characteristics feature. Lymphoblast is differentiating into two different types of cell, effector and memory. And then we're seeing the plasma cell, sorry, effector or plasma and memory cell. Then plasma cell responsible for secreting antibodies, and then we are getting the memory cell responsible for lifelong immunity to different types of pathogens. And now we're saying that how you can distinguish between these type of cells based on their cell membrane molecules. So as we are describing before, this is our one cell. Say for take an example. So we have different types of things inside the cell: nucleus, nucleic acids, etc., and different organelles, endoplasmic reticulum, Golgi bodies, mitochondria, ribosome, etc. Now, outside, this is the membrane. Okay, we can see the double lipid layer membrane. We have different kinds of proteins up there. So these functionizing by a PEG delivery agent, hydrophilic drugs, hydrophobic drugs, etc., etc., there outside of the cell. So based on that, whether these characteristics, this type of membrane molecules are present or not, we can say, well, what we are looking at, is this a memory cell, or this is a B small lymphocyte, or this is a T cell, or this is a NK cell, etc., etc., can be called. Okay. Now, all these chemical components has been grouped together and known and named as cluster of differentiation, or CD. That means I'm trying to say that, say, this is one type of cell, this is another type of cell, and this is another type of cell. So say this cell, this has a and a B and a C, three different types of components into their cell membrane. And this we have, say, C, D, and E. And here we have E, F, G, three different types of chemical components onto the cell membrane. So now all this together, we can say this is cluster of differentiation 1, or CD1. This is CD2, or this is CD3. So by seeing the presence of ABC, we can say that our cell is containing CD1. So all CD1 type of cells could be grouped together and say this is one particular type of cell. Fine. So now we will be focusing more on the T lymphocyte and B lymphocyte and how they are working together. First, B lymphocyte once again. From this point on, maybe it will be a little bit complex. So we have to understand the concept here. And once you will understand the concept, you will never forget in your entire life. Okay. So mature B cells. B cells developed, born or generated in the bone marrow, and then get matured in the bone marrow itself. They are distinguished definitely from other type of lymphocytes, such as T lymphocytes or NK cells or small lymphocytes, by display of one particular thing that is never present in any other kind of lymphocytes or other type of cells. That is membrane-bound antibody molecules, or immunoglobulin. So always we draw the antibody in a Y-shaped structure. We later will study the structure of antibody, but that's why it is said. So membrane-bound antibody. Only one type of cell that has the membrane-bound antibody. Do you remember that plasma cells were secreting antibody that was soluble? But here we are talking about membrane-bound antibody. All mature B cells has membrane-bound antibody. Now, one question may arise in your mind: what antibody you are talking about, and how antibody could be bound to this mature B cells membrane? Okay, because there are thousands and thousands and thousands types of microorganisms. So which antibody you are talking about? Okay, so I'm coming to that. Other type of molecule that expressed by the B cell membrane, so you can see different CDs are there. CD80, CD86, different. One particular thing that distinguishes B cell from other is called CD40. CD40 is always found on the B cell only. That's why you can replace the B cell and you can say CD40 cells. So in a scientific world, instead of seeing B cell, people say CD40 cell or CD40 plus cells, something like that. Another one is called class 2 MHC molecule. We will see about the class 2 MHC molecule maybe in the next class or next to next class. MHC means major histocompatibility complex. So three things we have found. One is membrane bound antibody. Another two is CD40 and class 2 MHC molecule. These are the distinguishing feature for the B cells. Okay, mature B cells, you can say. So I will come to this. Just give me 1 minute time. Okay, so this membrane-bound antibody or immunoglobulin molecules of the B cell, what are they doing there? They serve as receptor for antigen. Antigen means here microorganism, bacteria, virus, protozoa, etc. So you can see our green colored membrane-bound antibody is receiving an antigen. Or in other words, the antigen gets bound to this membrane-bound antibody molecule of the B cell. But the question still there, what is the source of the antibody and what is the antigen we're talking about, because there are several different kinds of antigen molecules are there. Okay, so now we'll see interaction between antigen and the membrane-bound antibody in a mature B cell, as well as interaction with the T cell, selectively induces the activation and differentiation of B cell clones of corresponding specificity. So maybe there are only 3-4 lines written over here, but we'll try to understand each and every word of these things and the whole concept. First of all, let's go back about this membrane-bound antibody. What is happening? So, as I said, the number of microorganisms are limitless. We honestly do not know that how many numbers of microorganisms are there. There's a different reasons for that, because the majority of the microorganisms cannot be cultivated in the laboratory. So we can feel their presence, but we cannot cultivate them and characterize them. That's a problem. Okay, so now what happens that mature B cells that are coming out of the bone marrow are not one type. They are also thousands and thousands of different types of B lymphocytes. What I mean by type? So they're all showing CD40, they're all showing MHC2 class molecules, and also they're all showing antibody bound to their membrane. But this antibody molecule is different in one cell to the another in terms of specificity. So say this is one B cell, this is another B cell, and this is another B cell. All of them has the membrane bound antibody. Okay, but this first one, the membrane bound antibody, is specific for one microorganism, say for Escherichia coli or E. coli. This B lymphocytes antibody is specific for, say, Salmonella typhimurium, responsible for typhoid. Okay, this one is specific for Streptococcus aureus, that gives us a Streptococcus infection, or Mycobacterium tuberculosis, causal organism of TB. So all has different types of antibody specific for one particular type of microorganism. Not only one, is actually, I should say, maybe you know, that one, as you have seeing in case of the COVID, that they have so many different variants are coming up. Similarly, say for even E. coli has 170 different types of variants. Salmonella have 150. So they are even specific to that level, specific to a particular variant. So when this Escherichia coli specific antibody bound B cell will find or come encounter to any Salmonella bacteria, this will do nothing. Salmonella bacteria will not be able to bind to here. This is not going to work because this is only specific for E. coli. So Salmonella will not bind here, will not bind here, but it will bind only here because this membrane-bound antibody is specific to Salmonella. So that is called the specificity that I was talking about, the attribute, specificity, diversity, memory, and self, non-self recognition. So specific. Now, due to different permutation and combination, the B lymphocyte we have with the membrane-bound antibody about anything in this universe is theoretically possible. So now, something, say from Mars, Mongol Graho theke, if some microorganism came into the Earth for the first time, okay, we have B cell membrane-bound antibody for that too. Okay, so even the microorganism that we're not thinking about, we're not aware about, not present in today's Earth, membrane-bound antibody molecules are still there. Why? Because antibody generates by a different mechanism, a different combination of mechanism in the genetic recombination thing. So they also do not have any idea whether it will be useful or useless. So if you have a mature B cells, an antibody which is supposed to bind to a microorganism which is not even present in this world, then the B cell will die after a certain time. It will roam around for a few days and it will die. It will be useless, but it's still there. If somehow that microorganism comes into play, then only those B cells will proliferate. If you are not very comfortable with the Mars example, I would say for example, that one particular disease, let's call the disease X. Disease X is actually endemic to a particular West African or Middle African country, Congo, just to take an example. Okay. So endemic means the disease only is found in that particular country, a particular region, not anywhere else in the world. So you are born and raised in India or maybe in the West Bengal region. So you never been into the Congo. So pretty sure that you never been exposed to that particular organism which is causing the disease X. But do you have the antibody? Do you have the membrane-bound antibody, B cells with the membrane-bound antibody against X? The answer is yes, we have. So it doesn't matter whether you are exposed to that and disease again or not. If I never been to the Congo again in my lifetime, maybe after attaining the age of 70 or so, I will die. No issue. Those B cells also will die with me, but that was there. So that is the thing about the B lymphocytes membrane antibody. Antibody about everything is present on their membrane-bound B cells, but they are not getting proliferated because they are not getting the chance to interact with those microorganisms ever. Okay. So if you have any doubt or anything, you can ask at any point of time. Now here, let's see what is happening. So this is our B cell. Okay. So B cell, we can see some membrane-bound antibody, and this yellowish one is a microorganism or antigen. So maybe two antibody molecules side by side, they formed a network, they bound with the antigen. Very good. That means we can say for sure that this red color antibody that I'm showing here must be specific for this antigen. Otherwise, they wouldn't have bind. They're specific to that yellow one. Then what they're doing? They are interacting with the T cell. As I said, the B and T lymphocytes, they work hand in hand in a cooperative manner. So the B cell is now actually interacting with a T cell. Let's not go into the nitty-gritty details at this point. Very simplified way I can say what B cell does. B cell actually presents the antibody molecule to the T cell. So you can see, sorry, antigen molecule. The antigen molecule is coming inside the B cell, and then the B cell using the MHC, major histocompatibility complex component, is showing the antigen to whom? To T cell, or one particular type of T cell known as T helper cell. So B cell, after internalizing the antigen, after binding the antigen through its membrane-bound antibody, internalizing it and presenting it to the T helper cell, asking, sir, can you please check whether this is an antigen or this is our own cell? So B cell will not take any decision. B cell may also bind to some of our own human cell. And if they present it to T helper cell for checking, T helper cell will say, no, this is not our antigen. This is our own cell. You do not need to take any action. Okay? But when B cell will actually, so you can see B cell means different types of B cell. Any B cell it could have. Maybe it bound to an E. coli, and then now it's showing it or presenting it to the T helper cell. Helper, that means it's helping. So if that is true, then T helper cell will say, yes, mate, it's time to take some action. This is indeed a microorganism. This is indeed a non-self thing that invaded in our system. How? T cell, B cell, they cannot talk like us, not in any human language, but they have a language that's called chemical language. So what they will do, they will release some chemical molecules, okay? Chemical molecules known as cytokines, etc., interleukin. So those chemical molecules will go into the B cell. Now B cell will understand, okay, my master here, T helper cell, has approved me, given me the approval to take some action. Okay, fine. Now what action it will take? The B cell will now be converted into two parts. Let's focus on the first part, to the plasma cell or effector cell. So B cell will be now converted to the plasma cell, and now plasma cell will secrete antibodies. So now you see these are the secreted antibodies, soluble in blood, soluble in lymph, not like this, not membrane bound, soluble. But all these antibodies are actually just replica of this membrane bound antibody. Why you need to have replica? Because otherwise they will not be able to bind to this antigen. This particular antigen, say yellow in color, is spread about different nook and corner of our body in different tissue space. So this soluble antibody will now migrate through blood, through lymph, to those spaces, and then it will bind and try to neutralize the microorganism or say the antigen here. Okay. So once again, what is the scheme? B cell at first binding to specific antigen through its membrane bound antibody, internalizing the antigen molecule and presenting some part of it to whom? To T helper cell or T helper lymphocyte with the help of MHC molecule. T helper cell, after recognizing it as a genuine foreign molecule, it's providing a signal to the B cell in terms of cytokine to convert to the plasma cell and to produce antibody molecule that will be called secretory antibody that is soluble in blood plasma. Those secretory antibody now came out from the plasma cell, will be distributed to different parts of the body, and anywhere they will find those particular antigen molecule, will bind to them and will try to neutralize the infection in such a way. But if this antigen or so-called antigen is not antigen that has been bound mistakenly by erroneous way by the B cell, then that will be rejected at this point and no signaling will be there. So B cell will not be converted to plasma cell. So no antibody molecules will be produced. But sometimes, due to some genetic error, this happens. Sometimes antibody molecules are produced against our own body cell. Maybe I mentioned that. So in that case, it's called autoimmune disorder. Autoimmune disorder is really a nasty thing, because nasty in the way that it's very difficult to treat, because your own system is kind of working against you. So, say for example, psoriasis, myasthenia gravis, such type of diseases are called autoimmune disease. Anyway, so this is the scheme: interaction between antigen and the membrane-bound antibody on a mature but naive B cell, as well as interaction with the T cell selectively induces the activation and differentiation of B cell clones of corresponding specificity. So here, the last line, I'm yet to describe a bit about this clone thing or selective differentiation, etc. So let's use a screen over here so that it will help for you. So as I was saying that we have millions of different types of B cell, difference meaning they have different kinds of membrane-bound antibody. Different kinds of membrane-bound antibody means membrane-bound antibodies are specific for one particular type of microorganism. So let's, for a simplification, say we have 4 different type of B cells with a membrane-bound antibody against, as I said, E. coli, Salmonella, Staphylococcus, Mycobacterium, etc., etc. Okay. So now, in the beginning, the number is very less. Okay. So 1 or 2, 1. So this is a 2 in number. There's also 3 in number, something like that. Now, one Salmonella bacteria entered into our system, Salmonella bacterium. So this guy at first rushed there, maybe tried to bind it and failed, because this antibodies for E. coli. This guy, Staphylococcus, also failed. So, okay, this Mycobacterium tuberculosis specific antibody is containing. So also no chance. Here also E. coli, because it's very specific. So only the Salmonella will be able to bind to this particular antibody, bound to this particular B cell. Let's call this B cell as the X. We call it an X, and this Y, Z, and P, other cell. So now what will happen? It will present a part of that antigen to whom? To T helper cell, also known shortly as TH cell. Okay. So TH cell is now, after recognizing this, yes, it's definitely Salmonella. It's not our own cell. It cannot understand the Salmonella or not, but what we can understand that it's a foreign cell. So it will provide its that to go ahead, take some action, put some chemical signal. Now, what will happen? So this particular cell X that we called will proliferate in huge number. Within a very short period of time, we'll be getting thousands of cells, thousands of B cells, which are all this X type. We do not need the YZ or YZ or P type here. We only require the X type, because they are capable to bound to the membrane. They are capable to bound to the Salmonella. That is the infection we are currently having. So this will be proliferate. So that is called, that we call this all one type of B cells as a one clone. So that is called clonal expansion. Clonal expansion is happening. So within a very short period of time, few hours maybe, started from 1 or 2 or 3 cells, that particular clone will be proliferated into thousands to control the situation, because there is not only one Salmonella. Maybe somehow with a food or something like that, you have consumed thousands and thousands of Salmonella, and you are going to get sick. So that to combat that situation, you have to have huge number of B cell which can bind to those Salmonella. Now what is going to happen? Huge number of X type of B cell, all with a membrane bound antibody, can bind to the salmonella. So each and everybody is binding the salmonella through their membrane-bound antibody. Each of them again presenting that to the T helper cell, and then after getting signal from the T helper cell, all of them are producing plasma cell. All of them are producing plasma cell, and plasma cell are producing secretory antibody. So within a short period of time, secretory antibody will be produced, and this secretory antibody will reach to this salmonella wherever it is in every tissue space. Okay, so that is the scheme how it works. So that is called clonal expansion. So what is happening? It's selecting one particular type of B cell. That is called clonal selection theory. Selectively induces and then activates, and differentiation of B cell is happening of the corresponding specificity. That means here specific to the salmonella. So that is the beauty of this whole thing of scheme works, especially for the humoral immunity thing. How two different types of B cell. We previously said that B cell is responsible for humoral immunity, T cell is responsible for cell-made immunity. But we have seen here that how these two types of cells are working in a cooperative manner. Okay, and how without the cooperation of the T helper cell, B cell is useless. Similarly, T helper cell also cannot do much if no antigen is presented to the T helper cell, and how the whole process is working and how the specific is. So here, once again, we can remember, we can think about the attributes that I've talked about the lymphocyte, that is diversity, specificity, memory, and self non-self recognition. So finally, in this process, a particular clone of B cell also divides repeatedly, and differentiates over a 4 to 5 day period, generating a population of plasma cell. I just mentioned that. So it will continuously divide, and will produce huge number of the same type of B cell just to combat, just to fight against one particular pathogen that we are concerned about at this moment. And secondly, it will also produce the memory B cells or memory cells. So memory cells, they will not produce any secretory antibody. They will just sit over there for a long period. What is the reason? As I mentioned, as I give you an example about the party crasher, that for the first time somebody crashed your party, you are a little bit confused. That is he actually any of my friend or friend's friend or something like that? Or really is he actually crashing my party? So it took some time for you to take some decision. So in between, maybe that particular person did some nasty thing in your party, they misbehaved with some of your guests, or and consumed some of your food and drinks, etc. But you memorized his face. Next time, maybe after 3 months or 4 months of time, when the same person tried to crash your party once again, this time you will recognize them because that person then and will kick him out. So in this way, you can protect your guests, you can protect your resources and food and drinks, etc. So that is also the reason for memory B cells. Next time, if the same antigen comes into the picture, comes into our system, then memory cells are already there. They will be able to bound to those cells and will be converted to the plasma cell producing effector antibodies. The entire action will be much more quick. You do not need to go through the same process once again. Okay, so that's why memory cells are important. And by using a vaccine, what we're trying to generate, we're just trying to generate these memory cells. It's not possible to have the soluble antibody in our system after certain time. So if I check, I do not have the we do not have any antibody against mumps, any antibodies against measles or rubella, something like that, because I vaccinated in the early days of my life. Okay, but if I get infected all those organisms, I have my memory cells. So memory B cells will be able to recognize those pathogens very quickly within a very short period of time and will take a very prompt action. So I will maybe feel sick for 1 day or two and then I will recover. So that is our whole confidence about the vaccination thing. We'll try to learn the vaccine thing much later point if we have anything that time. Okay. So we know that plasma cells synthesize and secrete antibody and all clonal progeny from a given B cell. So there are some biological term I'm using. I hope you're more or less familiar with this type of thing. Clonal progeny means the cells which are coming from a particular clone. I just said the clone was X type of B cell, one clone, one jati. So all the progeny, all the B cells that are coming out of the X is actually just like X. Okay. So all of them have the capability to show the membrane-bound antibody just like the cell X. So progeny from B cell secrete antibody molecules with the same antigen binding specificity. But these plasma cells are terminally differentiated cells. That means they have no longer will be converted to any other type of cell. They'll be there, they'll be produced, they will secrete antibody molecules, and then they will die within 1 or 2 weeks' time, and a new set of plasma cells will be come into effect if necessary. Okay, so these are this is about the whole thing what we have planned to discuss today. So as I said, a little bit maybe a bit complex. So now give your feedback. So if you have any questions or any problem in understanding anywhere, go ahead. No question? More or less you understood whatever I said? At least give some shout. Otherwise, how I know that whether you are sleeping or not here? Say something. Still cannot hear anything from your side. So fine, I'm just sharing the attendance form. Do the needful and then you can leave. Just give me a couple of minutes and try to share it. I have sent the attendance link. So provide your attendance, and that's all. See you in the next week.
Thank you, sir.
Here.
Thank you.
Thank you, sir.